Hi all, welcome back to Bio 148. Today we're going to be talking a little bit about, the birds and the bees. We're going to be talking about fertilization. Okay, so we're going to start by playing with meiosis and different ways that we can get the different gamete combinations that we can have in the sperm or the egg. And then we're going to be putting them together to fertilize, remembering how the cell then goes through mitosis to divide, and then it continues to divide to make our entire bodies, right? And there's changes in gene expression in those different cells that make it so that we have eyes, so that we have toes, and everything in between. Okay, so let's start by playing with some examples of some genes and on different chromosomes. So let's play with our note. Okay, so today we're going to be on day 17 of notes. Okay, so day 17. Oh, my daughter had my iPad. Let's get this sticker again. Okay. So day 17. So we're going to be talking about more chromosome definitions. We're also going to be talking about meiosis again, about fertilization, and then remembering things about mitosis and gene expression. Okay, so we're going to be adding a lot to this. Okay, so let's start out this way. So we're going to play with some gametes. So let's play with some gametes here. And this is going to be in a germline cell that's going to make some eggs, okay? So let's have a few different things going on. Let's have curly hair, and let's make this person heterozygous. So remember, heterozygous equals 2 alleles for a gene, okay? 2 different alleles, I should say. Let's do that. Oops. 2 different alleles, okay? And then we also have homozygous, okay? So let's say that this C, okay, like big C, little c, and let's say that this equals curly hair, okay? And in order to have curly hair, you only need 1 copy of the dominant, okay? So this is the dominant trait, okay? And the dominant allele, and this is the recessive allele. So as long as you have 1 capital C, you're going to have curly hair, okay? And then for the other gene, let's do, let's do hair color. So brown, let's do big B, and let's do little b, okay? Where big B is going to be equal to brown, okay? All right, so here we have a person that's heterozygous for both of these 2 genes, for curly hair and for brown hair, okay? Okay, so now I want to know what different combinations she can make for her gametes. So here we're going to first go through S phase. And when we go through S phase, remember that we're going to copy that single molecule to make a copy so that we have sister chromatids that are identical. So here we've got big C, and on the other, we've got little c, okay? We've got big B, and little b, okay? So here's after S phase, and at this point, actually, if I add this little line, I'm already in metaphase 1, okay? So you can go through all the phases like we did before, but sometimes it's nice to just kind of cheat your way through and be like, okay, I'm going to just look and see what happens with my gametes at metaphase 1. So here I can go all the way through all of meiosis 1, okay, and end up after meiosis 1 with something like this. Big B's, little c's in the other one, and little b's. Okay, so here's after meiosis 1. So remember, if we put that line down for the metaphase plate, it's going to be splitting the sister chromatids. Okay, so here's metaphase 2. So I'm already, remember here, and as a haploid, right? So don't forget, up here in metaphase 1, I was diploid. For all of meiosis 2, I'm going to be haploid, okay? These are going to split apart. And in my egg cells, I'm going to have the big C, big B from this one. Little c, little b. And note that I'm doing this little line across my C. That's to help me keep track. So I do this little trick where I put a slash through the lowercase when I'm dealing with anything that has like the uppercase and lowercase are the same, like C's, S's, P's, right? Anything that the uppercase and the lowercase are going to look the same, I always use that little slash, okay? V's, right? W's, right? Always use that. That's just like a trick, okay? All right, I'm just gonna get rid of that because we don't really need it. Okay, I need to make that smaller. Okay. All right, so now let's play with this. We have big C, big B is one option. Little c, little b is another option. But remember that they didn't have to line up this way on metaphase, at metaphase 1. So here's another option of what could have happened, right? So they could have lined up instead so that the little c's were on the left, right? And still, let's keep the big B's on the left. And then that means that the big C's are on the right. The little B's are on the right. Okay, so here I am in metaphase 1, but in a different germline cell, right? So here's option 1 on the left, and then option 2 is going to be here on the right. Okay, after we go through all of meiosis 1, so I'm just going to split this. Okay, after we go through all of meiosis 1, we're going to have the little c alleles in the left cell with the big B alleles. And in my other cell, I'm going to have the big C's and the little B's, okay? After we go through meiosis 2, where the sister chromatids are going to divide. So remember, homologous chromosomes divide during meiosis 1, right? Homologous chromosomes are dividing in meiosis 1. Sister chromatids divide during meiosis 2. We're going to get little c, big B, little c, big B, and this cell over here is going to produce big C, little b, big C, little b, okay? So I can get from this other way of lining up the homologous chromosomes on that metaphase plate, I can get little c with big B, right? Both of these two cells both have that. And I can get big C with little b, okay? So the 4 different gamete options that I can get are big C with big B, little c with little b, little c with big B, and big C with little b, okay? Okay, so let's just say like maybe this was Sophia again, right? So you guys did Sophia's gametes, right? And now if you guys have done the course pack at all, or you took this class before, you also might remember George. So let's say that George and Sophia have a baby, okay? And in George's cells, he has straight hair and he has brown hair, okay? And he's heterozygous for the brown hair, okay? So he has straight hair, so he has little c equals brown, okay? Oh, sorry, not brown, straight. Just getting ahead of myself. Straight. And he has big B, little b, so he has brown hair. Okay? Okay, so now let's see what different types of gametes he can make. So if we go through S phase with him, we're going to copy the C, and it's going to be little c on both because that's what he had to start with. And then he had big B, and he had little b, right? And so if here we're in metaphase 1, these are going to split apart after cytokinesis 1, so that we have 2 cells, one that has the little c's and the big B's, and the other one has the little c's and the little b's, right? Then in metaphase 2, these are going to separate during meiosis 2 so that we get big C, or sorry, little c, big B, little c, big B. Let's give these sperms some tails. Little c, little b, and little c, little b. Okay? So these are the sperm that George can provide. Okay? So up here, note that we had 4 different genotypes that that Sophia could provide, right? Down here, even if we were to flip this around, right? So if we were to flip this around so that the B's had switched, so we had little b's on the left. Note that here you still have that little c, little b option and the little c, big B option, which is the same as what we get down here, right? So down here for my sperm cells, I can have little c with big B, or I can, and that's present in both of these too, right? Or I can have little c, little b, okay? So in genetics, we'll talk a little bit more about crossing over and some different ways we can recombine things. But in this course, I want you only to worry about independent assortment and the chromosomes line up in different ways at metaphase 1, okay? So Sophia could give all 4 of these options, okay? So let's just grab one of each of these. Actually, I'll just make them down here. So she was able to do all 4 of these. So we'll take that and copy it. And bring it down, okay? Oh, did I give a word for this up here? I don't think I did. So when George was little c right here, okay? Little c, when he had the same allele for both, okay? This is called homozygous, okay? So homo meaning the same, right? So both alleles are the same, okay? So this could be little c, big C would also be it, right? Big B, anything that where the 2 letters are going to be the same is going to be homozygous, right? Versus up here we had heterozygous, right? Hetero meaning different, where we had those 2 different alleles, okay? So George was homozygous for the texture, for straight hair, and then he was heterozygous for hair color, okay? Okay, so let's come back though to these different eggs and these different egg options. So here I have, oops, I want these to all be pink. Okay, so here I have my different egg options. Let's get rid of that. So let's draw them out. So we have big C, big B. Here we have little c, little b. Here we have little c, big B. And here we have big C, little b. Okay, those are the 4 different gamete options that I could have. Now we could have either of these 2 sperm options that combine with it. Okay, so here are my 2 sperm options. So maybe the sperm that comes on here, attaches here, is big C, big B. Okay. And what will happen is this genetic information will go into the cell, okay? And that's fertilization, is when we have that genetic information going on. So you can imagine this sperm option coming into any of these, right? So let's copy that. Oops. So this sperm could go into any of these cells, right? So it'd be in different fertilization events, but it's possible. Any of these are possibilities, right? And so this will lead to a fertilization event, right? Oops. The egg is going to actually stay pretty much exactly as it is, okay? So let's grab all these eggs. Okay, I'm gonna put them down here. Get rid of those arrows, okay? Because the the sperm DNA is actually in there. So it was little c, big B. Little c, big B. Okay, so little c, big B. So this is when we have a zygote, okay, is when we're back to being a diploid, right? So here I have my zygotes, okay, and remember that zygotes are diploid, right? They have one copy from mom and one copy from dad, okay? So we have homologous chromosomes. Remember that these chromosomes, right, are not going to attach to each other, that they could be the same alleles, like here I'm heterozygous, or they can be the same alleles, so here I'm homozygous, okay? So all of these are going to be diploid and have homologous chromosomes, right? My current count for chromosomes is 4, and my DNA molecule count is also 4, right? So here are all my different options if this is the sperm that came in. But that isn't necessarily the only sperm that could have came in, right? So the other option here, let's copy these again. Oops, wrong thing. So let's copy all of my eggs. Hope I didn't capture some of those. Okay, big C, little c, big C, big C. Okay. So here I had that big C, big B. I had little c, little b. I had little c, big B, and I had big C, little b, right? So I'd already checked out this option, right? Where my sperm could have been little c, big B. Now let's play with this other option. So little c, big B, okay? So my sperm option here would be little c, wait, what did I say? Little b. Okay, little c and little b, right, is my sperm. So that is my other option here, and I'm just gonna add it to all of these. Okay, and so when we make these zygotes, right, we're going to be adding these together. So we're making these zygotes. Oops. Actually make this last one probably a little bit bigger. Okay, so little c, little b is this, what I got from the sperm. Little c, little b, little c, little b, little c, little b, little c. I might make that one bigger. Little c, little b. Okay, so these are my options if the sperm that was added was little c, little b. Okay, so these again are all zygotes. They're all going to be diploid, right? These homologous chromosomes are going to stay separate from each other, okay? So I'm going to have 4 chromosomes in each one. And I can also figure out the genotypes for all of these different ones. So in this first zygote, right, I've got big C, little c, big B, big B. And the second one, I've got little c, and then I've got, what is that one? Isn't that supposed to be little? Yeah, it is. Okay. Little c, big B, little b. Then I have little c, big B, big B. Big C, big B, little b, right? So I can see all my different genotypes. Big C, B, big B, little b. And this one's all little c's and little b's. Little c's, big B, little b. I'm gonna go inside, somebody's mowing. And then the last one was big C, little b, little b. Okay, so these are all my different options for my my zygotes to be made, okay? So here I've got 8 different options when mom was heterozygous for both genes and dad was heterozygous for one of the genes, okay? And we're gonna play next time, actually on Friday, with how these can can be used in a Punnett square instead, okay? But sometimes people just really like to walk through it this way first because it clicks a little bit better for them, okay? So here I've got all my different gamete options from mom and from dad, and I can look at all my different genotypes. And then from there, I can figure out what those phenotypes would be. So for this one, big C equals curly, right? I just need one of the big C's. And then big B equals brown, right? So this offspring would be curly and brown. This offspring, it's got homozygous little c, so straight and brown. This one is homozygous little c again, straight and brown. And this one's heterozygous, but it has one of the c's, so it's curly and brown. If I go down to the next set, when I had the other sperm as an option, here one big C is curly and one big B is brown. Oops. Here I had all little ones, so I had straight, and let's say little b and little b equals blonde, right? This next one is going to be straight and brown, and this final one is going to be curly and blonde. So you guys can see all these different phenotypes, right, that are dictated by the genotypes. And you guys can also tell me things like, oh, this individual here is heterozygous for hair texture and homozygous for hair color, right? This next one is homozygous for hair texture, it's straight, and little c equals straight, and then it's homozygous for the brown color, right, for hair color. So you should be able to tell me the different gametes that you can make, right? And you can do this through this process here of like, okay, here's option 1 for how they could line up on metaphase plate, or here's option 2 of how they could line up on the metaphase plate, or there's a little bit of a trick that you can use using some math, okay? So we're going to foil it. Do you guys remember foiling it? So I'm going to just draw a little line. And I want you guys to be able to foil this, okay? So we're going to go back to the pink. So remember mom had big C and little c, right? And mom also had big B and little b, okay? So to foil it, remember, we're going to do first, outside, inside, and then the last, okay? So first, outside, inside, last. So first, we're going to do big C and big B. So they're the first ones in the parentheses. Big C, big B, okay? Outside, here, big C, little b. Inside, little c, big B. And last is going to be little c, little b, okay? So these are the options that we could get from Sophia, right? Because Sophia was heterozygous for both, right? So she had big C, little c, big B, little b, heterozygous for both, okay? So these are the 4 different options that we could get from her gametes. They couldn't all happen in the same germline cell without any crossing over, but we're not going to talk about crossing over, okay? So this would have happened in, could have happened in 2 different gametes, okay? Or germlines, cells going through meiosis. George had something different, right? So George, let's see, let's remember, did he have straight hair, right? Oh, so George had little c, big B, little b, okay? So little c, big B, little b. Little c, remember he had straight hair, big B, little b, he was heterozygous for the brown color gene, okay? So he only can give the little c allele, but then he can give big B or little b, okay? So I can do big C with big B, right? So big C with big B, or I can do little c with little b. Oop, let me do that. Little c with little b, okay? So these are the only 2 options that the sperm can have, right? And that's what we had seen before. Versus Sophia had 4 different options because she was heterozygous for both, okay? So just kind of keep that in mind, that we, whether you're homozygous or heterozygous, is going to depend on how many different gamete options you can give, okay? Okay, so I want you guys to really be playing with this idea, okay? And actually, let's go ahead and introduce a Punnett square. I think this is a good place. So here, right, I have my 4 different gamete options for Sophia. So big C, big B, big C, little b, big B, and little c, little b, right? So these are my 4 gamete options for Sophia, right? You can even do the circles for the eggs, okay? And then from George, we can get little c, big B, or we can get little c, little b, okay? And we can only get those 2. Okay, so before when we were drawing it out, we were adding the sperm to the egg, right? So here we had sperm added to egg, right? And the egg became fertilized and turned into a zygote. So here we have our different egg options, again, across the top, okay? Across the top, so this is what we got from mom, okay? And then down the side, we have our different sperm options, okay? Sperm options down the side. So when we're trying to make zygotes, we want to combine the eggs and the sperm, okay? So I like to actually go this way where I just drag everything down, okay? So big C, big B's. The next one is big C's, and then it's little b. So I'm going to put the little b over to the side just so that it's out of the way for any other, anything else that I might want to put in there, okay? So big C, little b, and then I'm going to put the little c here in the middle, right next to a big B, little c, big B, little c, little b, little c, little b. So you can see I kind of left some space here for the other allele that I'm going to get from the sperm. I left some space here in the middle for the one that I was going to get from the sperm. Here I left it on the like edges, and then here I left it as like the first allele potentially, okay? And that just makes it so that I can easily see dominant and recessive things, but you don't have to do this, okay? You could instead put things always like this, where you have big C and then a blank, and then a big B and then a blank. Here I could have done big C blank, little b blank, okay? So it's up to you how you want to do it. I like doing it this way because then I can easily see what's dominant and recessive. Okay, so now I'm going to drag everything across. So I'm going to drag that big little c and the big B all the way across. So little c, big B, little c, big B, little c, big B. And then I'm going to do little c, little b for that second row. So little c, little b, little c, little b, little c, little b, little c, little b. Okay, so these gamete options that we're coming up with here are exactly the same as the ones that we saw here, okay? So I can find this little c, actually I'm going to change colors. I can find this little c, big C, little c, big B, little b, so it's heterozygous individual for both. I can find it down here, so it's right here. Oops. Right here where I have that combination of the big and the small, okay? So all 4 of these options that I found up here, right? So here, my zygote options, I had 1, 2, 3, 4 zygote options when I had the one type of sperm, and then another 4 down here. So total of 5, 6, 7, 8, right? Different options that I made kind of longhand by adding the sperm and the egg together. I did that here as well, right? So 1, 2, 3, 4, 5, 6, 7, 8. Note that these ones are not going to be the same as my ones up above, okay? Just because of the way that I had added them together. But just note that there's 8 zygotes possible, right? So 8 zygote options possible. Okay. And you can also take this and figure out what the genotypes are, right? Or sorry, the phenotypes are. So we have the genotypes, and now we could also add in what the back. Okay. We could also add in what the phenotypes are, right? So just like what we did before. So here I'm going to have curly because I have 1 big C and brown. Here I have curly and brown, again, right? Here I have straight and brown. Here I have straight and brown. Then down here I have curly and brown. Again, I have curly, but now I have blonde. I have straight and brown, and I have curly and blonde. Oh, that's not curly. Sorry, that's straight. Okay. Okay. Straight and blonde. Okay, so these are the same options that we saw before. We can do lots of things where we're figuring out genotype ratios and phenotype ratios as well. So maybe we want to know how often we get the curly and brown. Maybe we want to know how often we get straight and brown. How often we get curly and blonde. And how often we get straight and blonde. So for this one, the curly and the brown, I need one big C, and then the other one doesn't matter. And I need one big B, and the other one doesn't matter. For the straight, I need little c's only, and then a big B, whatever. So this could be a big B or a little b, right? It doesn't matter, okay? For this next one, curly, I need a big C, and then whatever. This one too. And then for blonde, I need little b, and then for straight and blonde, I need little c, little b, okay? So the dominant phenotypes, right, the ones with the capital letters, are going to happen more often. So here I see I have 1, 2, 3 in this case that are curly and brown, okay? For straight and brown, I've got, maybe I'll switch it. For straight and brown, I have 1, 2, 3, right? For curly and blonde, I have 1. And for straight and blonde, I have just 1 as well, okay? And then I can add these up, 1, 2, 3, 4, 5, 6, 7, 8. And that's how many zygote options I have possible, so I've checked that is correct, okay? So here I had 3 out of 8 were curly and brown, 3 out of 8 were straight and brown, 1 out of 8 is curly and blonde, and 1 out of 8 is straight and blonde. So I was able to come up with the phenotype ratios, that's what these are called, phenotype ratios, using my table here. I could have also figured all of these out by using my zygotes that I drew out up here, okay? I could also go back up there and look. So what color did I do the curly and brown? Was blue. Okay, so let's find all the curly and browns. Okay, so there's 1, 2, 3 curly and browns, which is what I had down here, right? So you can kind of check it with your zygote check, okay? Next I want straight and blonde. So, oh wait, straight and brown. And I did that with brown color, so let's switch it. Okay, so straight and brown. There's 1, 2, 3 straight and browns, and that's what I had down here as well. Then I had curly and blonde. So I'm looking for curly and blonde. It's here, okay? And then straight and blonde was my last one, and it was right here, right? Straight and blonde. So I can do it either by using a Punnett square here, okay? Punnett square. Or I can draw out the zygotes, okay? And both options are perfectly fine to try to figure things out. The zygotes will end up taking you a lot more time and is a lot more likely to kind of mess up. But as you are learning, I want you to think about that. The other thing I want you guys to remember is that here in my egg options, right, these are always going to be haploid, remember? So I'm going to always need 1 chromosome that has the C gene and 1 chromosome that has the B gene, okay? I don't want to see, like, I don't want to see this, okay? Don't give me that ever, okay? Don't ever, ever, ever give me that, okay? You always are going to want one of the C and one of the B, okay? I also, if I'm talking about two different genes, you need to give me both of them in each cell, okay? So each egg is haploid, but it has all of the non-homologous chromosomes, okay? So keep that in mind. Okay, I'm going to take a little pause to get some water, and then let's go over some learning objectives for today.